Hello, my name is Leslie Chong. I'm the CEO and MD of Imugene. It's a great honor to speak to you all today regarding the company. And my disclaimer. So Imugene as a snapshot, our market cap is $860 million. However, I think we're a little bit higher today. Our goal is to get over $1 billion, of course. Cash runway as of September last quarter was around $163 million. So it's quite a wonderful runway to develop our 4 platform technologies. We have a cellular therapy as well as an oncolytic virus and Oncolytics. I'll go into that in just a little bit. And our B-cell immunotherapy cancer vaccines. We have an in-house GMP cellular therapy manufacturing facility in Durham, North Carolina. And we are in solid tumors as well as now well into blood cancers, diffuse large B-cell lymphoma specifically. We have 5 ongoing clinical studies that we have reprioritized and, of course, long life patent portfolio. So with me today, we have our clinical development team. All of us have a wealth of experience developing specifically oncology drugs within CAR-T as well as immunotherapies. So we all have roughly about 150 years of experience together. It sounds very old, but, we're kind of young and feisty. We have 13 FDA clinical drugs that we've helped market, and that is all in oncology. 4 unique platforms, so I'll go right into those now. So Azer-cel CD19 allogeneic CAR-T. Just this morning, the company that we acquired this CAR-T from, Precision Biosciences, was able to out-license Azer-cel for non-cancer related diseases. So that helps us validate Azer-cel, and also the wealth of clinical data, I think, helped to out-license that. So we're quite proud of our acquisition. And just briefly, the difference between autologous CAR-T and our allogeneic CAR-T here, autologous, highly patient-dependent, where a patient comes in and sits there for roughly about 6 to 7 hours, getting leukapheresis where their T cells are taken out, and then re-engineered. It takes anywhere between 19 to 42 days, and during that time, patient oftentimes need bridging therapies. It's a laborious process as well as high manufacturing cost, and there's a variability in potency and due to the health of the patient's own T cells. Whereas Azer-cel, our allogeneic CAR-T, we take universal healthy donors, supersizing their T cells, and then we're able to ship these in batches, frozen to clinics and hospitals, and each batch can provide a drug for anywhere between 10 to 15 patients on demand when the patient needs it, and they are not waiting or need of bridging therapies. There is a promise of more efficient and cost-effective manufacturing, and also because it's healthy donor cells engineer for potency and persistence. We have a wealth of data after we've acquired Azer-cel from Precision Bio. 84 patients were treated already on Azer-cel, with 61 in non-Hodgkin's lymphoma. In the non-Hodgkin's lymphoma, we saw 58% overall response rate, and then with 23 patients in leukemia with 61% overall response rate. But I want to focus on the lymphoma patient because this is where we see opportunity in a registrational strategy. So out of the 61 non-Hodgkin's lymphoma patients, 18 were autologous or auto CAR-T failed or relapsed population, especially in that late line of lymphoma, the DLBCL population, a whopper 83% overall response rate, so which gives us a great opportunity for a registrational strategy. And the reason why is DLBCL is a great unmet need. Once they obtain second-line therapy, there is not a lot of therapeutic options for these very unmet need population, and this is exactly where Azer-cel could fit in. So first line, if you are diagnosed with DLBCL, you get a combination chemo as well as steroids. Roughly about 40% of that population fell off those or relapsed, and then they go on to further chemo as well as stem cell, and then now all the auto CAR Ts that are approved are moving fastly into this space, and there is nothing once you fell off of auto CAR T. And what we're finding is that roughly anywhere between 60 to 70% of the population that gets auto CAR T fell off those. So they relapse, and that creates a nice situation for our Azer-cel. So we know that even once a patient has received a CD19 therapeutics, once they fell off that CD19 marker is still relevant in 85% of that population. 60 to 70% of the population, once they received auto CAR T, fell off those. And by 2025, the use of auto CAR T in this particular space will grow by fourfold, thus creating a huge market opportunity for us. We've already had official and non-official meetings with the FDA regarding our registrational study. We will go into the relapse auto CAR T DLBCL space with roughly about 35 sites, including those sites that are currently participating in our phase 1B. So our oncolytic viral therapy. So in the simplest way, CF33, our oncolytic viral therapy, works. It has an extreme hatred for solid tumor. It only likes to infect it, and it only likes to replicate in solid tumor. So once it replicates, it then explodes the cancer cells and imparts that love onto other solid tumors. We know this happens because we currently have a phase 1 study with multitude of solid tumor. We have 4 arms in this study: intratumoral, intravenous, and then IT and IV in combination with pembrolizumab. We're roughly at a highest dose at this time in cohort 5, and we've already have some interesting response rates that I want to talk to you about. So at this time, at the data cut, this is October of 2023, the swim lane and the waterfall plot. I want you to focus on the red and the green. Those are sort of our mid-level doses, and yet you see a long longevity of persistence of response rate, especially in this one patient who had been on the study for more than 430 days with complete response and complete remission. And the waterfall plot is only getting more further down as we get higher in the dose. So in the red and the green, you see stability of disease as well as a clearance of solid tumor. So what we're finding is that the higher dose patients achieved a significant and durable reduction in their tumor burden. So that's really nice to see. And one in particular, this complete response or complete responder in the bile duct cancer type, patient came in, had failed or blew through 2 lines of therapy prior to coming on to our study. At the second scan, because their immunotherapy was activated or the tumor was turned hot, you see an explosion or increase in their tumor size, and by the third scan, decreased size, and by their 4th scan, the tumor is gone. And as I said, this patient remains a complete responder for the last over 430 days now. We love our CF33, and we know that CF33, our oncolytic virus, does in fact only target solid tumors. So we have a very clever way of allowing CD19-directed target to work in combination with our oncolytic virus. So oncolytic virus infects tumor cells, shuttles up or covers the solid tumor in CD19, and then you combine it with a CD19 therapeutic target. And this is exactly what happens with our Oncolytics, in the way that our viral infection happens quite swiftly, shuttling up a CD19 in combination with the CD19. In particular, we have a study with blinatumomab or Blincyto. That combination releases more viruses, shuttles up a CD19 and then combines with the CD19 therapeutics. You've got endogenous T-cell recruitment. You've got the viral load exploding. So it's a beautiful effect on the solid tumor. It's a way of making CD19 CAR-Ts work in solid tumor as a vehicle. So our phase 1 has already started. We call it OASIS. It's in combination with Blincyto, and we've already enrolled the first patient, so we are hoping to get data out there as fast as we can. What's so beautiful about these combinations is that there's myriad of CD19 directed targets that's already approved. So the combination is readily available for us, especially our CD19 targeting Azer-cel. So we've got a CD19 CAR-T that can combine with our own pipeline item Oncolytic. And we've done exactly that in a preclinical model. You see the combination, there was 100% killing of triple-negative breast cancer and gastric cancer within 72 hours. Is quite impressive, our initial preclinical work in this field. So we're hoping to progress this, again, as fast as we can. Our B-cell immunotherapy, this is our cancer vaccine platform, where in a phase 2 first-line gastric setting, we saw a hazard ratio of 0.558, quite impressive. And we're moving this into a phase 2 setting where it's fast to registrational strategy in second-line gastric cancer. We also have an anti-PD-1, and that this is a cancer vaccine against the PD-1 target. We have completed the dose escalation, and we saw impressive results with complete responder. There is a lung cancer patient in Australia, as a matter of fact, going on 3 years plus of complete response. Partial responder and stable disease were quite prevalent in this study. However, we're going to move this encouraging data into an indication we think that is quite thoughtful, the MSI-high colorectal cancer, and we've already started this study with 6 sites in Australia and then 6 sites in the UK. We have reprioritized our program so that we're prudent with our cash runway. So we have an allogeneic CD19 CAR-T that we're looking at blood cancer, DLBCL, and that auto CAR-T failure space. Oncolytic is the promise of making CD19-directed targets work in solid tumors. And then our CF oncolytic viruses keep producing wonderful data as we progress up the chain of the dose escalation. And our B-cell immunotherapies are doing quite well and moving right along. So our key catalysts for the next 12 to 24 months are pretty plentiful. We've got lots of Oncolytics news to be had with going into intratumoral as well as intravenous, and hopefully some expansions thereof. We've got Azer-cel, getting that DLBCL patients population. We have our Vaccinia study that keeps producing meaningful data for patients. And then in 2025, we'll move some of those into a phase 2, some registrational, some phase 2 in an expansion forum, and we hope to bring you further data as we progress. Our share price as of January 5th was at 12 cents, and our market cap, again, is $860,000. The majority of the shareholders are management and directors, as well as a few institutional holders like Vanguard and BlackRock. Why Imugene? I think the advanced portfolio, we have multiple shots on goal with our phase 1s and phase 2s. We have an extremely experienced management team with over 150 years combined experience in developing oncology drugs. We have ongoing clinical trials in blood cancers as well as solid tumors, and we have a robust cash runway. And we're pretty prudent with our cash. And so I'm back to the investment thesis that we have 4 platforms. 3 of them we have reprioritized. We have an in-house manufacturing facility. We are addressing areas of great unmet needs in blood cancers as well as solid tumors. And our 5 clinical studies are just doing that, proving that we can work in these very niche settings and long life patent portfolio. Thank you.